today's webinar discussion titled Cancer Cell Metastasis, Understanding the Process Using Insights from Single Cell Sequencing. My name's Pippa and I'm delighted to be introducing you to today's speakers, but before I do, I've got a couple quick announcements to make. Firstly, of course, to thank our speakers for today for joining us. Secondly, to encourage you to use the tab on the right-hand side of your screen to submit questions. You'll also be able to upvote other questions, highlighting the best ones to be answered by our speakers today. You can also use the chat function to interact with members of the audience, but please note that only questions asked on the question tab will be able to be directed to the speakers. Thirdly, the slides for today will not be made available, but you will be able to rewatch the discussion on demand if you miss anything. This will be available immediately after we finish today, so you don't need to wait to be able to re-access the webinar. We will be running a Q&A at the end of the live session, so please continue to ask questions throughout the presentations. And without further delay, I'm delighted to introduce our speakers, Lydia and Katarina, who will introduce themselves at the beginning of their presentations. So over to you, Lydia.
Yeah, thank you very much, Pippa, for the kind introduction and also for the invitation today. So my name is Lydia and I'm a senior scientist at the University of Cologne, and my expertise is the preclinical oncology, especially the different mouse models we have, and the cancer entity, which is most interestingly is lung cancer. And I will focus in the discussion of the metastatic cascades and the immune evasion mostly on lung cancer. So we will cover three different questions today. So why is the discovery of organ-specific metastatic tracks so important? What are possible ways of metastasis formation? And how can the formation of organ-specific metastasis be prevented? And first of all, you see here the typical sites of metastatic relapse of several leading solid tumor entities. And as I promised, lung cancer comprising many different entities, but focusing on lung adenocarcinomas and small cell carcinomas. The principal metastatic sites are the brain, the bones, the adrenal gland, and the liver. So the first reason why the discovery of metastatic tracks is important, because we have so many solid cancer entities that develop systemic organ metastasis. The second reason is that for the reason we have solid cancers with a really high metastatic potential, this is also the leading cause of the cancer-related death we observe in men and women. And unfortunately, the metastatic cancers are frequently diagnosed at an extensive stage of disease with many unresectable tumors. And this is also for the therapeutic outcome, of course, not beneficial. This is a study, the IMpower 133, where extensive stage small cell cancer patients have been evaluated. And here the researchers evaluated the efficacy and safety of atezolizumab, which is an immune checkpoint blocker of PD-L1, in extensive stage disease patients. And interestingly, they found that the presence of metastasis can be associated to a weaker therapy outcome under immune checkpoint blockade. So the next question we would like to address today: what are possible ways of metastasis formation that impact the response to immune checkpoint blockade? So we have, in general, in a simplified fashion, three different modes of metastasis formation. So we have the amoeboid mode, where the cell-cell contact is really weak. We don't have a cell to extracellular matrix contact, no proteolysis, but the cells are really motile and they find their path through the extracellular matrix to form distant metastasis.
Hi everyone, really sorry about our technical errors that we're having here. We're just going to see if we can get Lydia back on. So for the meantime, I'm going to bring Katarina onto the stage, who will start her Oh, I think Lydia's just joined us. Lydia, are you able to
Yeah, of course. Sorry. I
Okay,
don't know what happened.
So I'm going to bring back up your presentation, and you guys can start. You can start again whenever you're ready. Okay.
Yeah. So sorry for the that I lost the internet connection. I think it's the really bad internet situation in Germany. Yes, we were talking about the different modes of metastasis formation. So I already introduced you to the amoeboid mode, and now we will move on with the mesenchymal mode, because here we have an active kind of metastasis formation where the cells, the tumor cells really form contact with the extracellular matrix, perform active proteolysis, are really motile, and generate their path through the extracellular matrix, and also perform epithelial to mesenchymal transition. What came up in the last years and is really famous right now is the collective cell invasion. And there we have cell-to-cell contact, cell to extracellular matrix contact, proteolysis, and also a high cell motility. And we have a leader cell follower cell organization with a path generating mode where the tumor cells pull themselves through the extracellular matrix, and cells following these path generating cells. And we also see frequently partial epithelial to mesenchymal transition. So when we take a look at the cells, which are normally the leader cells, they can be either tumor cell derived, so that we have either hybrid epithelial mesenchymal leader cancer stem cells, or we have fully mesenchymal leader stem cells, or also basal cancer stem cells. But we also have possibly leader cells from the tumor microenvironment, so from the stroma. For example, tumor-associated macrophages or cancer-associated fibroblasts, which can also fulfill the function of leader cells in the metastasis formation. But yeah, no matter which leader cell in the metastasis formation process we have, all distant metastases need to fulfill a certain metastatic cascade, namely intravasation, extravasation, and colonization. And we will shortly look into this. So, I mean, step one of the metastatic cascade is clear. We have invasion. So tumor cells leave their primary site of tumor, but what happens next? So after this, we have the process of intravasation, where the tumor cells make contact with the endothelial walls and enter the bloodstream. Then here they need to survive in the circulation. And finally, when they enter their site of metastasis, they need to extravasate, form a pre-metastatic niche, and then colonize the foreign organ. And also, of course, need to survive there. And one thing I would like to highlight here, which might become interesting also later, are the integrins, because they are expressed on the tumor cells and needed to make contact with the endothelial walls. So yeah, now you are well equipped for our last question. How can the formation of organ-specific metastasis be prevented? So as I said, I will focus on lung cancer, and the following slides will show you some published data and also some new findings. And the reasons for choosing this are, of course, lung cancer is a cancer entity with systemic metastasis. And we have a really cool mouse model to mimic this disease, which forms autochthonous lung tumors and metastasis, which is driven by conditional RB1 and TP53 knockout. This was published many years ago by the group of Anton Berns at the Netherlands Cancer Institute. And yeah, this mouse model closely mimics the human disease. And interestingly, also in SCLC, SCLC, small cell lung cancer, we know that there are different subtypes within this tumor entity, and these subtypes also differ regarding the immune signatures and also regarding the features of epithelial to mesenchymal transition. So what we did, we took the mouse model, inhaled the mice with adenoviral Cre recombinase, which then generates the conditional RB1 and TP53 knockout. And after a certain period of time, mice start to develop tumors, which we can monitor by micro-CT and MRI. And then we treated the mice with the atezolizumab analog, so anti-PD-L1 monoclonal antibodies. And we wanted to improve this therapy by combining an anti-angiogenic therapy to this immune checkpoint blockade, because we wanted to achieve a better infiltration of immune cells into the site of the tumors. And upon this therapy combination, we assessed progression-free and overall survival, and these Kaplan-Meier curves show the monotherapies compared to the combination therapy in red. And you see that we had a synergistically improved progression-free and overall survival upon combining anti-VEGF to the anti-PD-L1 therapy. And importantly, this combination was even better than the standard chemotherapy for this tumor entity, cisplatin etoposide. But how can the combination of anti-VEGF-A to the anti-PD-L1 do this? So first of all, we analyzed the tumor microenvironment, and we saw that we had an improved immune cell tumor cell ratio upon the therapy combination. And we really see an increased T cell infiltration into the tumors. But most importantly, we also identified an acquired resistance mechanism to the anti-PD-L1 targeted therapy, which was driven by an exhausted T cell phenotype indicated by double positivity for TIM3, which is another immune checkpoint receptor, and PD-1, which is the corresponding receptor to the PD-L1. And here you see when we combine the anti-VEGF-A to the anti-PD-L1 therapy, we can rescue this exhausted T cell phenotype. Most importantly, we have also analyzed the tumors currently responding to the combination therapy and tumors progressing under the therapy. And here you see that the tumors that acquired resistance to the anti-PD-L1 therapy are the ones where we see the exhausted T cell phenotype. And this was not present at the limited stage of disease. And we also had matched liver metastases to the primary tumor, and also in the liver metastases compared to the primary tumor, the exhausted T cell phenotype was more pronounced. We wanted then to show the direct effect of VEGF signaling on T cells, and for this purpose, we analyzed patient-derived PBMCs and stimulated them with VEGF in vitro. We compared hereby patients who received radiochemotherapy or radiochemotherapy plus immune checkpoint blockade, and there we see that upon VEGF stimulation, that we have also the induction of an exhausted T cell phenotype. So we ask in the next step, where does the VEGF-A comes from, which drives the exhausted T cell phenotype? So to answer this question, we generated CRISPR-Cas9 mediated VEGF-A knockout cell lines, and we injected them subcutaneously to see if there's a direct effect on the tumor growth. And this is not the case for small cell lung cancer. And after this, we autotopically injected this VEGF knockouts versus the wild type control autotopically into the lung. And surprisingly, these tumors did not respond to the anti-PD-L1 therapy we gave. So the tumor cell-derived VEGF-A was not the responsible one to reverse the T-cell exhaustion. But when we analyzed the VEGF-A knockouts more deeply, we saw a really huge invasion of the ribcage by tumor cells, and we also detected liver metastasis. And then we asked ourselves, how can the blockade of VEGF-A, VEGFR signaling in the tumor cells can mediate this invasive phenotype? And to answer this question, we again inhaled mice driven by RB1 and TP53 knockout, waited for tumors, and then generated different cell lines from this and performed scratch assays and also transfer assays. And there we saw that with prolonged VEGFR inhibition, the cell migration was increased. And we also found that the prolonged VEGFR inhibition increased the epithelial to mesenchymal transition and also the focal adhesion kinase signaling, which is a really important pathway in cell migration. And we also saw an upregulation of CD29. Now you may ask, well, what is CD29? It's an integrin, so namely integrin beta 1. And integrins classically form heterodimers and are prominent for cell migration, cell adhesion, survival, and proliferation. And we then analyzed publicly available data sets of SCLC patients and also human SCLC cell lines. And there we saw a clear correlation of integrin beta 1 or CD29 with vimentin, which is the classical mesenchymal marker. And then we wanted to see if CD29 is really a driver of the invasion in SCLC. And to answer this question, we also generated cell lines from our murine model, from our murine lung cancer model. And we generated cell lines from different lesions of the lung. And there we have seen that there seems to be a population which is CD29 low and CD29 high, which was quite interesting. And we then performed again scratch assays and transfer assays. And here stimulated with fibronectin, which is the canonical ligand for integrins. And we have seen that CD29 expression correlates with the fibronectin-induced migration. But is CD29 truly required for the migration? In the next step, then, we knocked out CD29 in our murine SCLC cell lines and performed again the scratch assay. And we could see that CD29 knockout fully abrogates fibronectin-induced cell migration. So finally, to also check if this integrin signaling blockade might reduce liver metastasis in vivo, we again inhaled our mice, started the serial micro-CT, and once there was a measurable target lesion in the lung, we started to block the integrin signaling axis, harvested tumor material after a certain period of time, and performed immunohistochemistry analysis of the harvested organs. And there we could see that the integrin signaling blockade significantly reduced the liver metastasis formation. So to sum up my talk, you have heard our points concerning three different questions. So why is the discovery of organ-specific metastatic tracts so important? What are possible ways of metastasis formation? And how can the formation of organ-specific metastasis be prevented? So there were four main reasons for the metastatic tracts because the solid tumors develop distant metastasis. And these metastases are related to increased mortality rates in men and women worldwide. But unfortunately, the metastatic cancers are frequently diagnosed at a late stage. And last but not least, the metastases are associated to a poor therapeutic outcome under immune checkpoint blockade. And ways of metastasis formation, so the partial epithelial to mesenchymal transition and the collective invasion are really prominent. And I think for small cell lung cancer, at least, the way to go. And the metastatic cascade, nevertheless, has to be fulfilled and finished, namely extravasation, and colonization. And this seems to involve the integrin signaling. And finally, the blockade of the integrin beta-1 dependent signaling can abrogate the metastasis formation and may be also a suitable therapy combination for immune checkpoint blockade for extensive disease patients. So thank you very much for your kind attention, and sorry for the bad internet connection, and it was a pleasure, and I would like to hand over to Katarina.
Thank you very much, Lydia, for the nice talk. So my name is Katarina Pankova. I'm a senior product manager with Singleron Biotechnologies, and I'm very happy to be here this afternoon and have this opportunity to present to you Singleron's case study on breast cancer metastasis, as well as our solutions beyond traditional single-cell RNA sequencing. So let me jump straight into single-cell RNA sequencing and what it actually is and what advantages it has in cancer research and in studying the metastases in general. So traditional bulk sequencing has been tremendously useful to characterize gene expression in the tissues of interest. However, the problem arises if you want to study tissues composed of mixed cell populations. So in such cases, bulk RNA sequences only gives you the average expression profile of the entire sample. And basically the differences in gene expression between the individual populations of cells that constitute this tissue gets lost. So for this reason, single-cell sequencing technologies have been developed. So the single-cell sequencing methods take individual cells as an input and are able to provide transcriptome information for each individual cell separately. So basically, using single-cell sequencing, you are able to get precise expression profile for every single cell type that is present in your sample. And also, I would like to emphasize here that even though the name says single-cell sequencing, the output is not the transcriptome of single cell, but rather the transcriptome of thousands or tens of thousands cells that you get in one experiment. So this method at the moment is a really high throughput, and we are talking about tens of thousands cells processed simultaneously in a single experiment. So how do we enable for this high throughput single cell sequencing? Singleron's technology is based on around the scope microchip. So this is a simple device that is shown on the upper left part of the image. It can be easily held in hands and operated either manually with a pipette or using our specialized instrument. So basically, this microchip is a microfluidic device that is covered with microvels, and we load single-cell suspension onto this microchip, and the individual cells get separated into the microvels. After that, we introduce barcoding beads. Again, one barcoding bead falls into each microvel. And ideally, we end up in a situation where you have a single cells and a single barcoding beads in the microvels. So afterwards, we introduce lysis buffer. The cells get lysed, and the mRNA that they release gets bound to the barcoding beads. We retrieve the barcoding beads from this microchip, and we proceed with the reverse transcription, cDNA amplification, and the library construction. And basically, the result is a sequencing-ready NGS library, which is compatible with the Illumina instruments. So our barcoding beads are covered with oligonucleotides that have poly-T sequences. Basically, this is how we capture the mRNA. And what is also important to say about these barcoding beads is that each of the barcoding beads carries a different cell barcode, so a different sequence. And basically, this is how the beads can be distinguished one from another, because they have different cell barcodes. And in the end, when we do the bioinformatic analysis, we can assign transcripts that have the same cell barcode. We know that they originated from the same microvel, and therefore they belong to the same cell. So what kind of data can you get using our technology, and what kind of insights can you get on your cancer research or the metastatic process in general? So I'm going to present a study that was published in the journal Oncogenesis approximately half a year ago, and one of our colleagues from Singleron is a co-author on this paper. So the paper deals with the transcriptome profile of the cancer cell lymph node metastasis. And the key research questions that this paper is aiming to answer is the comparison of the cellular composition and the transcriptome profiles between primary tumor and the lymph node metastasis. And the second question that the paper is dealing with is the characterization of the breast cancer stem cells. So the design of the study is depicted here. So basically the samples from 5 different breast cancer patients were collected. For each of these patients, molecular subtype of the breast cancer was determined using the classical pathological examination methods. And from each patient, in total, 3 samples were taken. So 1 from the primary cancer site, from the breast, and the 2 from the axillary lymph nodes. And all these samples were processed using our technology. Basically, the single-cell suspension were partitioned on our microchip. The libraries were created, and after sequencing, the bioinformatic analysis took part. So in the end, the data comprised of gene expression information for more than 96,000 individual cells. So how this data looks like can be shown in this plots here. So let me briefly walk you through what you are looking at. Let's start with the graph on the very left side of the slide. So we are looking at the UMAP projection of the single-cell RNA-seq data. So basically, every single dot that you can see on this graph corresponds to a single cell. There is more than 96,000 dots corresponding to more than 96,000 individual cells that were actually processed in this experiment. And you can see like this, many of these dots actually overlap because there's so many of them. So as a result of the single-cell RNA-seq experiment, you get the expression matrix. So basically the gene count for all protein encoding genes basically that you can look at in your experimental system. And what the UMAP projection does is that it reduces the dimensionality of the data such that it plots cells with a similar gene expression in the cluster, so close to each other. And the cells that have different gene expression are further apart on these plots. So just to sum it up, every single dot is a single cell. Cells with similar gene expression are clustered together on these UMAP plots. What you can see in colors here are basically samples coming from 5 different patients. So basically, every single patient is assigned a different color in this plot on the very left side. Now, if we move to the middle portion of the slide, we are looking at the very same data set. So basically, the same clustering of the single-cell RNA-seq data. This time the data is colored based on the origin of the sample. So in gray, you can see the samples that come from the primary cancer, so from the breast. In the green, you can see the samples coming from the lymph nodes that were positive for the cancerous cells. And in the purple, you can see the cells that come from lymph nodes negative for the cancerous cells. And lastly, in the right portion of the slide, you are looking again at the very same clustering of the single-cell RNA-seq data. This time the colors correspond to the individual subtypes of the cells that are present in your samples and they're identified. So authors in this paper were able to identify 18 subtypes of the cells in total, including ones that are rather rare, such as, for instance, the dendritic cells or the cancer stem cells. Now, once you have your single-cell RNA-seq dataset, it's a lot of data that you have on your hands, and you can choose to plot them in a different fashion to basically get various insights on different processes or properties of the samples that you are basically looking into. So one of the things that you can do with this data is to look at the cellular composition of the samples. So on this particular plot, the authors split the samples into two main groups. So the samples coming from the triple-negative breast cancer patients and the non-triple negative breast cancer patients. And within these two groups, they created three subgroups. So the samples coming from the primary cancer versus the samples coming from the lymph node positive for cancerous cells and the lymph nodes negative for the cancerous cells. So the colors on each of these 6 bars show you the proportion of the individual cell types that were present in these samples, and the list of these 18 cellular subtypes that were detected. In this study is basically shown on the right side. So we can already see that the samples of different origin have different kind of cellular composition, and this is one of the advantages of the single-cell RNA-seq, that it enables you to understand the details of what is in your sample. The authors also look into more detail into the breast cancer stem cells that are basically involved in the metastatic process. So on the left side, you are again looking at the UMAP plot of the single-cell RNA-seq data. Again, I want to emphasize here that this is a dimensionality reduction of the data. So for every single dot, for every single cell, you have the whole gene expression matrix with gene counts for all protein encoding genes, but you don't see this data on this plot. You only see a single cell as a single dot and the cells that have similar gene expression clustered together. So this time the authors only took the cancer cells and they reclustered them, hoping that they would be able to basically get specific groups of cancer cells that would have different gene expression and would differ in function. And this is actually what they were able to achieve. So within the 9 clusters that they identify, they were able to show in the middle part of the image how the gene expression of the marker genes specific for each cluster actually differs. So on this heat map, on the x-axis, you can see in color 9 different clusters that were identified in this analysis. And on the y-axis, you are looking at 8 top differentially expressed genes that are marker genes for each cluster. And what you are trying to see in this plot is the step-like structure that would basically validate your approach and confirm that these 9 clusters differ based on the marker genes which were identified for each of them. So the breast cancer stem cells, I believe, are the cluster number 6. So they have a defined set of marker genes based on expression of which they differ from the other clusters. And using them, the markers reported previously in the literature, such as CD44 or the ADLH, the authors were able to identify this cluster as the breast cancer stem cells. The further analysis that was performed of the breast cancer stem cells involved the analysis of the copy number variants as well as the trajectory analysis, but I won't have time to walk you through the details of the data. And I would like to also mention the insights that you can get on the function of the immune system in the metastatic process, and how you can use single-cell RNA-seq data to characterize the transcriptome of the immune cells and basically draw conclusions on or implications for their function in the metastatic process. So in this particular plot that we are looking at, the authors looked at different types of immune cells. So they are listed on the left side, left part of the figure. So they basically look at the CD4 naive T cells, CD8 effector T cells, B cells, macrophages, Tregs, and so forth. And they basically plotted how the gene expression between two different samples differs in this individual population of immune cells. So basically, if there's an certain upregulation of the high number of genes in a particular immune cell type, this would result in a large red bar for the particular comparison. And if there's a large number of genes downregulated in the particular comparison, this would result in a large blue bar. So basically, this large number of upregulated genes, you can take it as an approximation for the activation of the immune system. So if you, for instance, look at the right portion, or the most right column of this image, you can see the comparison between the triple negative breast cancer and the samples from the primary cancer versus lymph node negative for cancer cells. And you can see that in primary cancer, there is higher activation of the first 5 groups of immune cells depicted as the larger red bars. So basically, single-cell RNA-seq data enables you to understand the activation of the immune cells that are present in your sample, and basically provides you more insights onto the tumor microenvironment and how it changes. So what this study was able to show using single-cell RNA sequencing, so as I mentioned, study was able to provide transcriptome profiles of more than 96,000 individual cells from 15 paired patient samples. It is the first study describing transcriptome profile of breast cancer lymph node metastasis using single-cell RNA-seq. The study provides characterization of the breast cancer stem cells, and it also provides insight into the breast cancer metastasis mechanisms. Now I would like to talk to you briefly about what you can do with single-cell RNA-seq data that is rather non-traditional and is beyond the widely used single-cell RNA-seq. So for those cancer researchers that are interested in the function of the immune system in more detail and would like to understand the sequences of the TCR and of the receptors at the T cells on the T cells and the B cells, we offer immune VDJ profiling. The rationale behind it is that if you use single-cell sequencing, 3' based, in general, you have very good gene coverage at the 3' ends of the genes. But if your sequence of interest, such as, for instance, the C3R3 region on the VDJ sequences, if this region lies further away from the 3' end, you need an additional solution. And this is exactly what the immune VDJ profiling can help you with. Basically, we have dedicated beads that are designed to capture not only the whole transcriptome, but they also have probes to capture the constant regions of the TCR and the BCR receptors. So basically, at this very first stage of capturing of the mRNA, we already enrich for the TCR and BCR sequences. And later during the library amplification process, we perform another additional 2 rounds of the PCR enrichment to make sure that you get sufficient coverage of the TCR and BCR sequences that are of your interest. The second solution that we offer, and maybe especially of interest to the cancer researchers, is the Focuscope kit. So the Focuscope kit is basically, the logic behind is the similar as for the VDJ profiling. the single-cell sequencing is very good at giving you high gene coverage at the 3' ends of the genes. But if your sequences of interest, the mutations or the gene fusions lie further away from the 3' end, you basically need to make sure that you get sufficient coverage at this point as well. And for this reason, we offer, again, specialized beads that not only capture the whole transcriptome using the poly-T sequences, but they have also probes specific for the sequences of your interest. And these probes can also help you to capture the viral sequences, for instance, if this is something that you are interested in, or you can design your own customized panel of 5 to 8 genes that basically you can study and apply to your research questions. So the good news for those of you who studied the lung cancer, we have already on the market lung cancer druggable mutation panel that with beads which have probes dedicated for the capturing of the genes that are relevant for the lung cancer researchers. And soon we will have out also the breast cancer panel and the blood cancer panel. I also want to emphasize here that we are still open for any customized solution. So if you have your own panel of 5 to 8 genes which you would like to study in more detail, it's certainly possible from our side. Lastly, I would like to briefly mention Singleron's service that we offer in our service lab in Cologne, Germany. So our workflow starts with the tissue or the single-cell suspension. Then the single-cell suspension we load on the microfluidic chip, and then we create NGS library, which we send for sequencing. And lastly, we also perform bioinformatic analysis. So basically, you can choose if you want to get our kits and basically do this workflow yourself in the lab. Or for those of you who don't have interest in this or capacity to do so, we offer a one-stop shop solution for single-cell sequencing, where you simply send us your tissues and we take care of the rest. And we even do the bioinformatic analysis for you and produce publication-ready data. So what is our trick? How we actually make sure that the samples that you send to us are of high quality. So we have a so-called tissue preservation buffer. This is a special solution in which you can store your biopsy tissues for up to 72 hours. So you don't freeze the tissues, you store them at 4 degrees. And we have data showing that there are minimal changes to the transcriptome or the cell viability while you store these tissues in our preservation buffer. So this solution is very customer friendly. So if you are interested in our service, we simply ship you tissue preservation buffer. You drop your biopsy samples in this buffer and send it to us. Tissues can be stored in this buffer for 72 hours, which is sufficiently long for samples from anywhere in Europe to send to our lab in Cologne, Germany. Also, I would like to mention that we have a lot of experience processing different types of tissues. So we have been working on more than 3,000 different research projects. We have served more than 600 different customer organizations. And as an interesting fact, I would mention that there are more than 200 million of single cells that we have already sequenced. And our focus is on the human and the mouse data. As for the different types of samples, we have experience with various tissues, more than 400 different tissues, including ones that are difficult to process, such as, for instance, the brain tissue, bone tissue, or the fat tissue. And lastly, our overall success rate for processing of the customer samples is currently at the 94%. So this is it from me. Thank you very much for your attention. I would be happy to answer any questions that you might have at the moment. If you have any questions in the future, feel free to contact us. Here is our email address. We will be happy to help you with any inquiries that you might have.
Thanks, guys. I think they were some really good presentations. I just wanted to thank you again for your time. So now, yeah, just as we're going to move on to our questions, which will be moderated by Katarina herself. So please continue to keep sending questions into the question section at the live form feedback. And yeah, so over to you, Katarina.
All right, so let me start with the first question on the bioinformatic analysis. So the question is whether if we do the bioinformatic analysis in-house as a part of our service, whether we still provide the raw counts file and matrices to the researchers. Yes, we provide you with the FASTQ data and basically also the analyzed data. That's no problem at all. So raw data, providing of the raw data is no problem. We do it on a regular basis. There's another question regarding the barcodes that we used in our technology. So the question is, let me briefly. Okay, so question is on the UMIs, so the unique molecular identifiers that are present on our beads. So the unique molecular identifiers are basically pieces of it's a short sequence that differs on every single oligonucleotide that is attached to the barcoding bead. And basically we use it to count how many transcripts belong to every single gene. So basically the UMI count is how we count the transcripts that belong to every single gene. And yes, they are. So every single UMI that is attached to the barcoding bead is different. So this is how we make sure that we are able to count the transcripts. I think this would be a question for the Lydia. So if the metastasis features depend on the primary organ, is it possible to determine the primary tumor site from the metastasis?
Yeah, that's a good question. In general, it's possible. So this is also routinely done in the pathology. For example, the features of the keratin cytoskeleton are kept frequently in the metastasis. And when the physicians identify a possible site of metastasis, they order pathological stainings for the cytokeratins. For example, cytokeratin 8 and 18 are the cytokeratins particularly expressed in the lung. And if there is, for example, a liver tumor and this expresses the cytokeratins, then they are quite sure that the origin is from the lung. But there are also other specific transcription factors that are also kept in the metastasis.
Thank you. Another question for you. Do you know what are the current trends in new directions in the field of metastatic cancer research and how it will look like in the future? And maybe how single-cell technologies may be helpful in that regard?
Yeah, definitely. So I think analyzing matched samples from metastasis and the primary tumor, no matter if they originate from the liver or from the brain and then from the lung or from the breast, for example, the single-cell sequencing technology is the technology to go. Also to check for targetable targetable proteins for personalized medicine. But not only this, so I think also the identification of metabolic changes and epigenetic markers will be important in the future. And the more or the broader the view we get, the safer we are in the decision for therapy.
Mm-hmm. We also have a question on the throughput of the single-cell sequencing technology that the Singleron actually offers. Basically, the throughput of that we normally offer for the standard chip with this 150,000 microvels is up to 10,000 cells that you can basically sequence in a single run. We also have dedicated solutions for the even higher throughput. We call it high density microchip. And using this microchip, you can process up to 30,000 individual cells in a single run. So this would be to give you a brief idea on how many cells can be sequenced at what costs. We had a question also for Lydia. The question is whether there is a known mechanism for the T cell response to VEGF-A WRT exhaustion.
So the VEGF-A with regard to T cell exhaustion, yes, right. It's known since, I think it was 2015, that VEGF-A is in general able to regulate different immune checkpoint receptors on T cells. And we also have asked ourselves, how can VEGF do this? I think it's a transcriptional program in the T cells that is switched on by till now not known mechanism, which is delivered by the tumor cells to the T cells. But we also have checked when there is T cell exhaustion, if the VEGF receptors are expressed. And after a certain period of time in acquiring the resistance, there is upregulation of the VEGF receptors. But when you check initially, the T cells do not express the VEGF receptors. And I mean, there are also different ones. And in our case, it was upregulation of VEGFR1, which we have seen on the T cells, and which is the likely driver of the exhausted T cell phenotype independence of VEGF-A. But what is the internal molecular mechanism in the T cells? I think this is not known so far.
I have another question for you regarding the metastasis in liver. So the question is whether you have assessed the presence of metastasis in the liver using immunohistochemistry.
Yes, so we did a bunch of immunohistochemistry analysis in the liver, also for all the different epithelial and mesenchymal markers, because we wanted to see if we have a pure mesenchymal metastasis or if it's a partial EMT. And for the reason that the SCLC at least is neuroendocrine differentiated, we checked also for these markers, and we can say that the liver metastases we see are partial EMT, so expressing both markers, referring to the collective invasion. But I would like to mention here that when you look for metastasis, it's always important not only to look for isolated metastasis, but always in correlation to the primary tumor. Because when you look in one patient or in one mouse and only look for the isolated metastasis, then you cannot really see the what is the change compared to the primary tumor. So the individual levels of certain marker expression in the metastasis, for example, in the liver, can be really different. So it's always important to have a reference material from the primary tumor.
Mm-hmm. Thank you. Another question would be whether there is a time dependence for blocking integrin signaling to present further metastasis.
Yes, so I think this is really the case. We haven't tested yet. But of course, it's the question whether the blocking of the integrin signaling can also help when the metastasis is already formed, or if we prevent that the tumor cell leaves the primary site of the tumor and do the intravasation step. So this remains to be tested.
There's another question on our product focus scope, which I can take. So the question is, how many different targets can be added to the focus scope assay? So basically, our recommendation is that is between 5 to 8 genes. So this can be basically sequences that you are interested in, which you simply submit to us. We will develop custom-made beads for you, validate the assay, and within a range of few months, you get the ready product for your ready customized product for your research questions. A question for Lydia. What's the most relevant thing at molecular level to check the metastasis mechanism?
I would say the different transcription factors driving the epithelial to mesenchymal transition, because they are also expected to drive which mode of metastasis formation is chosen. For example, the amoeboid mode is closely connected to the STAT3 and NF-kappa B signaling, whereas the partial epithelial to mesenchymal transition and collective invasion is more linked to the ZEP1 snail slug twist transcription factors. So I think checking for this would be most suitable.
Another question for you, Lydia. So, what are the what are the potential drawbacks or issues when introducing combined ICD and anti-integrin therapies clinically?
Yeah, the largest drawbacks, I would say, for lung cancer at least, that there are known toxicities. Yeah, for the other cancers, I mean, the combination of antiangiogenic therapy and immune checkpoint blockade is already in clinical trials, for example, in clear renal cell carcinomas and I think also in NSCLC. So I guess the largest drawback will be also in this combination the toxicities, which are also known from the chemotherapy immune checkpoint therapy combination. Yeah.
Another question to maybe go deeper in this direction. So, are VEGF inhibitors already widely used in lung cancer treatment?
Yeah, they are. So the former ones like sunitinib, for example, are known for years for non-small cell lung cancer, and also targeting the VEGFR2 receptor by monoclonal antibodies is also in clinical trials. So it's really on vogue.
Maybe the last quick question. So, what determines the way of metastasis formation? When it does choose amoeboid, mesenchymal, or collective ways to metastasize?
Yeah, well, I think this is the direction where my other answer already pointed in. So it's the transcriptional network that is switched on in the tumor cell. Of course, what drives the different transcription factors is always the question. Yeah, if we could answer this, then we can cure cancer. But I think if you want to know what drives the metastatic mode, then you can check for the transcription factors. And if you want to know where the metastasis might go, then you may look for tumor cell metabolism, because it's also known that that different organ tropisms are caused by the metabolism of the tumor cells.
Thank you very much, Lydia, for the nice talk and responding to all the questions that came up for the audience. And I think I will hand over to Pippa.
So I just want to say, I think that's all we've got time for today. Thank you again to everyone who's joined us for this discussion and presentations. Don't forget that you can watch the webinar back on demand anytime using this exact same link. Again, a final thank you to Lydia and Katarina. I think we can all agree that they've done an amazing job today, especially with answering the questions that have come through. So have a great day, everyone, and we'll see you again soon. Thanks.
Thank you.
Thanks.